Hello students, welcome to my channel Vihira Academy. I am your educator Dr. Heena Devnani, and today we are going to discuss chapter number 3 of our 12th standard biology textbook, that is human reproduction. This is the third part of the chapter. In the first part we have studied about male reproductive system. In the second part we have studied female reproductive system, and today we are going to do the third and the last part of this chapter, which include gametogenesis, fertilization, implantation, pregnancy, development of the fetus, parturition and lactation. So let's start with gametogenesis. What is gametogenesis? It is a process by which the male sex organs and the female sex organs produce male and female gametes. That means the testis and the ovary produce sperms and ova. If the testis produce sperms, it is known as spermatogenesis. If the ovaries produce ova, it is known as oogenesis. So first we'll start with spermatogenesis. Here a chart of the entire process is given. If you remember this chart, no need to study the theoretical part. You can write in the exam very easily. So let's study what is this chart is all about. First, the spermatogenesis process begins with type A spermatogonia. You can also call them as the germinal cells or the stem cells. What they do? They start the process. Initially, they will carry out cell division. Here you can see one cell is there and so many cells have been formed. So a cell division occurs over here, and that cell division is mitosis. By mitosis, the spermatogonia produces many different cells. These are type A spermatogonia. By mitosis, they will produce type B spermatogonia. Now, what is mitosis? Mitosis is a type of cell division in which the number of chromosomes remain the same. Like if this cell is having 46 chromosomes, or we can say 23 pairs of chromosomes, these will also have 23 pairs of chromosomes. So type B spermatogonia will be diploid. This entire phase is known as the first phase of spermatogenesis, which is the multiplicative phase. After this, the type B spermatogonia will increase in size. They will increase in size and form primary spermatocytes. And the process by which they increase in size, or the phase by which they increase in size, is known as the growth phase. That is the second phase of the spermatogenesis. First phase was the multiplicative phase, in which type A spermatogonia were divided and they formed type B spermatogonia. Second phase is the growth phase, in which type B spermatogonia will form the primary spermatocytes. Now again there will be another division phase, which includes meiotic division. First meiotic division and second meiotic division. We have studied in 11th standard that meiosis occurs in two parts, meiosis 1 and meiosis 2. So first what will happen? Primary spermatocytes will undergo the first meiotic division and they will form secondary spermatocytes. Then the secondary spermatocytes will undergo the second meiotic division and they will form spermatids. Now, once the spermatids are formed, the process of division is stopped. You have to note here that these spermatids are haploid. The spermatogonia were diploid, type B spermatogonia were diploid, type A spermatogonia were diploid, but because of the meiotic division, the spermatids are haploid. And now the next phase of spermatogenesis comes, which is known as spermiogenesis. It is also known as the differentiation phase. What happens here? The spermatids get differentiated to form the mature spermatozoa. There are three types of changes that occur here. Initially, there will be changes in the nucleus. What happens to the nucleus? The RNA, the proteins, the nucleolus, all these things in the nucleus are of very less importance now. Only the DNA is important. So the DNA will increase in size and rest all will get degraded. They will reduce in size to reduce the space of to reduce the size of the entire sperm. Then what happens? Then there will be formation of this acrosome. This area is known as acrosome. The pointed area of the sperm is known as acrosome. There will be formation of acrosome with the help of proacrosomal granule. This proacrosomal granule will be formed from the Golgi complex of the cell. And the third step is formation of centrioles and the formation of tail. Here in the middle piece, centrioles are present. There will be two centrioles over here. Centrioles will divide into two. First will be the proximal centriole and second will be the distal centriole. The distal centriole will form the entire tail region of the sperm. Then in the middle piece there will be accumulation of mitochondria to provide energy. So these are some of the changes which occur in the spermatids to form sperms. That's why this process is named differently and it has been given the name spermiogenesis. Now when all this process takes place, some residual bodies are also released. So this is the entire process of spermatogenesis, a very important process of the chapter. There are some hormones which affect spermatogenesis. These hormones are known as gonadotropin releasing hormones. These hormones are secreted by the hypothalamus. Their level increase significantly during puberty. What is puberty? Puberty is the age where reproductive maturity is begin. The increased levels of gonadotropin releasing hormone will stimulate the release of two gonadotropins, follicle stimulating hormone and luteinizing hormone. What is the role of follicle stimulating hormone? It acts on the Sertoli cells and they stimulate the release of some factors which will help in the process of spermatogenesis. So when the follicle stimulating hormone is released, it will actually aid in the process of spermatogenesis. Luteinizing hormones will act on the Leydig cells and they will stimulate the synthesis and secretion of androgens. If you remember these Leydig cells, Sertoli cells, we have done in the male reproductive system. What is the function of Sertoli cells? They provide nourishment to the developing sperms. What is the function of Leydig cells? They release the androgens. So here androgens are released and these androgens will stimulate the process of spermatogenesis. So that was about spermatogenesis. Now let us study the structure of sperm. First of all, see the diagram. This is the diagram. This is the head region, the middle piece, and this is the tail region of the sperm. It consists of four parts: head, neck, middle piece and tail. Here it also has a neck. Plasma membrane covers the whole body of the sperm. The head portion of the sperm contains a haploid nucleus. Now you have studied spermatogenesis, so you know what is a haploid nucleus and how is it formed. The anterior portion of the head contains an acrosome. Acrosome is actually a structure that is filled with digestive enzymes. Which enzyme is present over here? That is your homework. Find it out and tell me in the comment section which enzyme is present over here. So acrosome is a structure that is filled with digestive enzymes that help in dissolving the membrane of the egg cell and it also helps in fertilization of the ovum. That we are going to see in the later part of this video. Then comes the middle piece. Middle piece contains numerous mitochondria. Why mitochondria are present over here? To give energy, to produce energy so that the sperms can carry out their motion. It is very important for the motility of the sperm. Sperms should carry out motion. Now, during one ejaculation, around 200 to 300 million sperms are released in a single ejaculation. Now, out of these 200 to 300 million sperms, 60% of them, maybe they have normal shape and size. And at least 40% out of these 60% should have vigorous motility. That means they should be able to move. Only then the fertilization is possible. Now, these sperms are not released alone. They are released along with some secretions of the accessory glands present in the male. So, along with the secretion, these sperms constitute a fluid which is called as semen. Semen is a milky white organic fluid which is released by the penis during ejaculation. What does this semen consist of? First of all, it consists of 200 to 300 million sperms. Along with this, it also consists the secretion of accessory glands. and ducts like epididymis, vas deferens, seminal vesicle, prostate glands, and the bulbourethral glands. The testicular hormones, that are the androgens, or we can also call it as testosterone, they actually maintain the function of all these male axillary ducts and glands. So this is the entire structure of a sperm. Head, here is there is acrosome, this is the nucleus consisting of DNA, this is the centriole, the neck, the middle piece consisting of mitochondria, and then there is the tail, which is also called as flagellum. The entire structure is covered by a plasma membrane. Let's move to oogenesis. Oogenesis is a bit complex than the spermatogenesis. Now what happens in oogenesis? There is development of oocyte, or we can also call it as egg. Along with the development of egg, there is development of the follicles also. So you need to study both of these together. Let us start with this. When a female is born, it already has lot of follicles which are called as primordial follicle. Primordial means primitive follicles are already present, consisting of oogonia. Now here what happens? Primordial follicle will consist of many oogonia, and these oogonia will undergo mitosis to form primary oocytes. Primary oocyte will just begin meiosis 1. You have to remember, it will just begin meiosis 1. During this time, there is no change in the primordial follicles. See, this is the oocyte inside. Here, the blue color is the nucleus, and it is covered with follicular cells. So, this entire structure is known as primordial follicle. The number is many. In a female, when she is born, the ovary consists of many primary follicles, primordial follicles. Now what happens during adolescence? During adolescence, the changes start in a female. Adolescence means the puberty. That is when the reproductive maturity is starting. At that time, changes start in a female. So what happens? The primordial follicles will get converted into primary follicles. You can see the changes in the cells. Initially there were follicular cells. Now these cells are known as granulosa cells. At this time, the primary oocyte will remain unchanged. Now what will happen? Slowly and slowly, the primary oocyte will complete its meiosis 1. One thing you have to remember here is during puberty, the number of oocytes get reduced to 400 to 600 oocytes, and every month only one oocyte is released. So only one oocyte will undergo this process, and this occurs every month, consisting of the menstrual cycle that we are going to see later. So what happens here? Every month one oocyte will complete its meiosis 1. At the same time, the primary follicle will be converted into secondary follicle. You can see here granulosa cells are present. Now the ovum also has a covering which is known as zona pellucida, and there is an external covering which is known as theca folliculi. Here meiosis 1 will be completed. Primary oocyte will be converted into secondary oocyte and first polar body. The first polar body is a very reduced cell. Now why this happens, why there is unequal division, this is your second homework for today. There is an unequal division. In the spermatogenesis we have seen that there was equal division. But here there is unequal division. One cell is very large and the other cell is very, very minute. And at the end this first polar body dies. Now at the same time what happens? Secondary follicle is converted to tertiary follicle. Tertiary follicle has a cavity inside it. This cavity is filled with a fluid, and this cavity is known as antrum. Here there is a space between the ovum and the antrum which is known as cumulus oophorus. Now the theca is formed into two layers, theca interna and theca externa. Here the secondary oocyte will get ovulated. Now when it gets ovulated, what happens? You can see here. The theca externa and theca interna will break and the ovum is coming out. At which stage it is coming out? It is coming out at this stage, secondary oocyte stage. This is the follicular fluid. There is some bleeding in the antrum, and this follicle, now a mature follicle, is known as Graafian follicle. So the graphene follicle opens up and the ovum is released. This process is known as ovulation, a very, very important process. Every month, only one egg is released in this way by the ovulation process. Now, when the ovulation occurs, the ovum is released, or we can say secondary oocyte is released. This follicle is converted into corpus luteum, and then it gets degenerated after a fixed period of time. Now what happens? Suppose if there is fertilization, then the secondary oocyte will meet the sperm. You can see here sperm is there. The secondary oocyte will meet the sperm and it will undergo second meiosis. When it forms second meiosis, at that time two cells will be formed. One cell will be the embryo and the second cell will be the second polar body, which will again die. Now, if the secondary oocyte is not fertilized, then what happens? The ovum will die. So this is the entire process of oogenesis. A complex process because two things you have to remember here. These two things go on simultaneously. Let's move to a very important process again, menstrual cycle. This is the reproductive cycle which occur in female primates like monkeys, apes and human beings. The cycle of events start from one menstruation till the next one. And this cycle is very, very essential for the production of oocytes. Along with the production of oocyte, this cycle also plays a major role in the preparation of uterus. The uterus prepares itself every month for pregnancy so that it can carry the pregnancy. This cycle is generally of 28 days, but it can go on up to 35 days. And one egg is released per cycle in a normal condition. Now what happens in this cycle? There is a phase of menstruation of around 5 to 8 days, and what happens during this menstruation? It is a process by which blood and the mucosal tissues will be regularly discharged from the vagina. This is also known as a period or monthly. Now when the menstruation starts in a female, that age or that phase is known as menarche. It generally begins at puberty. If there is menarche, that means the girl has entered into the puberty. And this age for menarche is different in different people. In some it can be 10 years, in some it can be 15 years, in some it can also go up to 17 years. Menarche signals the beginning of reproductive age in females. Then there is another phase which is known as menopause. Menopause is actually the stoppage or the cessation of the menstrual cycle. Why the cessation is there? Because there are no oocytes to be released now. As I said, there is a fixed number of oocytes. There are fixed number of oogonia in a female. Once these oocytes or these eggs are finished, there is menopause. There is no menstrual cycle then. This also varies from person to person. The average age is around 45 to 50 years. Now have a look at this chart because the entire cycle has been explained through this chart only. We'll start with this 0 days. As you can see here, some dot-like structures are drawn. What does this indicate? This indicates bleeding. So from 0 to 5 days, 5 or 7 days, let's take 7 days, what happens? There is bleeding in the uterus. Why the uterus wall gets bleeded? Why there is bleeding? Because the wall of the uterus gets shredded off. Continuously it is budded off and continuously there is breaking of blood vessels over here. That's why there is release of blood and tissues. At this time what happens? The level of ovarian hormones is very less. The level of anterior pituitary hormones is very less. The temperature of the body is little bit above 36 degrees Celsius. And at this time, the follicles are in the growing phase. Then there is the first phase of the menstrual cycle after bleeding, which is known as follicular phase. It generally occurs from day 16 to day 6 to day 13. What happens here? There is sudden increase in estradiol. The hormone from the ovaries, estradiol, it increases at a sudden pace. So what happens? The uterus wall, this is the wall of the uterus. The uterus wall also rises in size. The inner wall of the uterus, that is the endometrium. It increases in size. How does it increase? Here the blood vessels accumulate and the tissues are formed and new cells are formed. So this line increases. Level of ovarian hormone estradiol also increases. There is some increase in follicle stimulating hormone. If follicle stimulating hormone is increased, that means the follicles will also increase. Here you can see growing follicles. We have already seen primary follicle, secondary follicle, tertiary follicle, Graafian follicle, right? So till day 13 this happens. On 14th day there is release of ovum. Here you can see body temperature increases. Luteinizing hormone is at its highest peak. The level of estradiol is reduced. At this time ovulation will occur. Now after ovulation, what happens? The level of progesterone increase. Why this level of progesterone increase? Because of the corpus luteum. We know that Graafian follicle is converted to corpus luteum. This corpus luteum is responsible for increasing the level of progesterone, and this level of progesterone is important for increasing the size of the uterus wall. You can see there is a drastic increase in the size of the uterine wall. At this time, estradiol level also increases and then again decreases. Both the hormones of anterior pituitary are reduced. Body temperature rises in this entire period. Now when corpus luteum gets degenerated, it is converted to corpus albicans. So corpus luteum is degenerated, that means the level of progesterone will come down. As the level of progesterone comes down, the wall again breaks, and then again there is the menses. The luteal phase goes on from day 15 to day 28, and after day 28 again day 1 begins, that is menses. So this is the entire process of menstrual cycle. Let's move to the next step. Menstrual cycle occurs when there is no fertilization. Now what happens when there is fertilization? First of all, how does fertilization starts? It starts with insemination. What is the meaning of insemination? Insemination means during coitus or copulation, coitus means sexual intercourse, the semen is released into the vagina by the penis. Now in this semen there are sperms. The sperms are motile, so they swim rapidly through the cervix. They were released in the vagina. From vagina they enter the cervix. From cervix they enter into the uterus, and from the uterus they go to the ampullary region of the fallopian tube. You have to remember this, that fertilization occurs in the fallopian tube only. Now in the fallopian tube sperms have reached. At this time the ovum will be released by the ovary if the ovulation period is there. Fertilization will occur only if ovum and sperms are simultaneously transferred to the ampullary region. Otherwise fertilization will not take place. And what is fertilization? It is actually the fusion of the sperm and the egg. Now what happens? Suppose if the sperm has reached the ovum, these changes will take place. This is the ovum, this is the egg membrane, this is the pre-vitelline space, these are the granules. You don't need to study all this in detail. You just have to remember this is the covering of an ovum, which is also known as zona pellucida. Now what happens? The sperm has reached the ampullary region, the ovum has reached the ampullary region. So the sperm will come in contact with the zona pellucida. It has acrosome over here. Acrosome has some hydrolytic enzymes. This is the homework that I have already given you. You have to find the name of this enzyme. So the enzyme is present over here. What it will do? It will digest the wall of the zona pellucida. So here you can see it has digested the wall. It has reached the pre-vitelline space. Then it will fuse with the membrane, plasma membrane of the egg. And then the nucleus will enter inside. The entire sperm will not go, only the nucleus will go inside, and then it will fuse with the nucleus of the egg. At this time, the egg will undergo secondary meiosis to form an ovum. So this is the entire process of fertilization. After fertilization, what process takes place? Cleavage. Cleavage occurs. What is cleavage? Now here fertilization has taken place. One cell is formed, which is known as zygote. The zygote will undergo cleavage. Here from day 2 cleavage will start. First of all, from one cell 2 cells will be formed, from 2 4 cells will be formed, from 4 8 cells will be formed, and then 16 cells will be formed. When 16 cells are formed, it is known as morula. The zygote is known as morula. Then the morula will undergo a process of cavitation and it will form a blastula. Now this blastula, it has zona pellucida outside. This blastula, what it will do, it will break open the zona pellucida and it will be released. This process is known as zona hatching, because the zona pellucida is broken over here and the ovum, and the blastula has come out. Now what it will happen, this entire thing from here to here, this entire thing will occur from the fallopian tube to the uterus. The zygote will go on traveling from the fallopian tube to the uterus, and when it reaches the uterus, it will undergo zona hatching. On the 7th day, it will start its implantation process. These are the cells of the uterine epithelium, the uterus or the inner wall of the uterus, the endometrium. Now what happens? This outer layer, the layer which is shown in yellow color, this outer layer is known as trophoblast. So now what will happen? The cells of the trophoblast will digest the cells of the uterine epithelium and it will try to enter in it. When it enters in it, what happens? Slowly and slowly the uterus will take in the entire embryo, and it will try to cover it from all its sides. Here, this entire thing will go like this. A C-shaped covering will be formed. Then certain processes will go on. You don't have to go into detail. We have to reach over here. When this entire process of implantation is over, then what happens? See here, some finger-like projections will be formed from the embryo as well as from the uterus, and there will be interdigitation. What is interdigitation? If you take both of your hands and if you just combine both the fingers into it, you lock both the fingers with each other of both your hands, that is known as interdigitation. So here also same thing will be formed. Finger-like projections are formed from the embryo. Finger-like projections will be formed from the inner wall of uterus and they will undergo interdigitation. When interdigitation occurs, this region or this structure which is formed over here is known as placenta. Now between the placenta and the embryo there is a connection, a cord-like structure which is known as umbilical cord. This is the umbilical cord. This is the entire embryo. For your understanding, I have added a GIF over here. You can see the embryo has gone inside, it has been covered, and the finger-like projections increase in size. Let's move to pregnancy and embryonic development. After implantation, finger-like projections which are called as chorionic villi will appear on the trophoblast. Just now I told you the finger-like projections increase in size. These are known as chorionic villi. These chorionic villi will be surrounded by the uterine tissue and the maternal blood, and they will become interdigitated with each other and they will form a structure which is known as placenta. What is the function of placenta? The function of placenta is to form a connection between the mother and the fetus. Its role is to supply oxygen and nutrients to the embryo and to remove the carbon dioxide and excretory waste from the embryo, from the fetus, and send it to the blood of mother. It is connected to the embryo through umbilical cord. I have already shown it to you. The embryo transports nutrients and waste to and from the placenta through the umbilical cord. Placenta is also very important because it also acts as an endocrine gland and it is responsible for secreting some hormones such as human chorionic gonadotropin, human placental lactogen, estrogens, progesterone, etc. Now, HCG and HPL, human chorionic gonadotropin and human placental lactogen, are only released during pregnancy. That's why when these pregnancy kits, you might have heard about pregnancy kits which are available in the market, by which any female can check whether she is pregnant or not at home itself. These pregnancy kits consist of receptors of these two hormones. So when urine is released, these two hormones will be present in the urine and then they show the positive sign on the pregnancy kits. Now, along with these four hormones, in the later stages of pregnancy, another hormone which is called as relaxin is also produced. Relaxin is responsible for the delivery process. During pregnancy, production of various other hormones is also increased. These hormones include estrogens, progesterones, cortisol, thyroxine, prolactin, etc. High levels of these hormones are essential for supporting the fetal growth, metabolic changes in the pregnancy, as well as the maintenance of the pregnancy. And soon after the implantation, the embryo will differentiate into outer ectoderm and inner ectoderm. As soon as it gets implanted, it will form outer ectoderm, it will form inner ectoderm, and then the mesoderm will also be formed. These three tissues will together, outer ectoderm, inner endoderm, and then mesoderm. These three tissues will form all the tissues of the body, all the organs of the body. The inner cell mass also contains stem cells, which have the ability to give rise to all the tissues and organs of the body. Here we can see 9-month development of a child. This is how the child grows. This is 1 month, 2 months, 3 months, and so on. Let's see what happens. In first month, arms, tail, gill grooves are formed, webbed toes are formed. This is very interesting for you to note that in the initial stages of embryo development, the mammalian embryo, the avian embryo, that means embryo of the aves, reptilian embryo, amphibian embryo, and the piscean embryo, all these embryos look same in the initial stages. They have gills, they have tails, they have webbed toes. In the later stages, they change and get formed according to their classes, but initially all of them are same. This indicates evolution that we are going to study in later chapters. Let's move to second month. What happens in second month? The length of the embryo is around 1.6 centimeter. Weight will be 1 grams. Retina, eye cavity, fingers and toes, upper limbs, genitalia, and all these things will be formed. In the third month, length will be around 5.4 centimeter. Weight will be 14 grams. Nostrils will dissociate. Genitals and neck are formed. Sucking reflex will appear. It will appear as if the child or the embryo is sucking. After this, the embryo will be known as fetus. Till third month, it is known as embryo, then it will be known as fetus. Fetus means small human. So in the fourth month, it will appear like a small human. Its length will be 11.6 centimeter. Weight will be 100 gram. Heartbeat will be more pronounced. Cartilaginous tissues will stiffen, and major organs will be formed. In the fifth month, length will be 26 centimeter, weight will be around 300 grams. The movements will be more regular, and now the mother will be able to feel the movements. At this time, head and body hair will appear. Sixth month, length is 30 centimeter, weight is 600 grams. The eyelids the child will now be able to open its eyelids. Lungs and lung circulation will be developed. Heart circulation will be developed, not lung circulation over here. Seventh month, 37.6 centimeter. Weight will be 1 kilogram. Arteries and veins will be formed and fat will get deposited under the skin. At this time, it will appear a bit transparent and you will be able to see the arteries and veins. In the 8th month, the length is 42.4 centimeter, the weight is 1.7 kilogram. Development will almost be complete and fetus will be viable if it is born. Fat deposit will increase and senses will be developed. 9th month, size is 48 centimeter, weight will be around 2.6 kilogram. The child will be fully developed and it will be ready for delivery. You can see here, it has turned its head down, so the delivery will be from the lower part. Then comes parturition. Parturition means delivery. Now before parturition, you have to understand another term, that is gestation period. What is the meaning of gestation period? The average duration of pregnancy. It is known as gestation period. In humans, the gestation period is around 40 weeks, or you can also, in months, you can also tell it as 9 months. After 9 months, the parturition occurs. Parturition means the process of childbirth by which the fetus is expelled or delivered out due to vigorous contractions of the uterus. There will be contractions of the uterus because of which the child will be pushed downwards and it will come out from the cervix and the vagina. Now, how does this start? A fully developed fetus, if the fetus is completely developed, the fetus and the placenta will induce parturition. The fetus itself will give signals to our body that now it is ready to come out of our body. Now what happens when they give signals? At that time there will be mild uterine contractions. The uterus will start to contract and these will be very mild. So the mother will come to know that now the time has come. And when these mild uterine contraction starts, it is known as fetal ejection reflex. The fetus is ready to get ejected of the body. So these contractions will then trigger the release of oxytocin from the mother's pituitary gland. It will send signals to the brain and oxytocin will be released. Now what this oxytocin will do? It will stimulate more stronger uterine contraction. So the uterus will start to contract more. And when uterus is contracting more production of oxytocin will start. Again, when more production of oxytocin is there, again the uterus will start to contract in a very stronger manner. Again there will be oxytocin secretion and this process will continue till the contractions become very strong, so that the baby is expelled out of the uterus through the birth canal. And as soon as the fetus is delivered out, the placenta also comes out of the body, and at the same time there is release of relaxin. Relaxin is a hormone which relaxes the uterus muscles because it has undergone many strong contractions, so now it needs to get relaxed, and therefore the relaxin hormone is released over here. Have a look at this. This is the onset of the labor. This is the uterus, the pubic bone, the bladder. Here there is the vagina, rectum, cervix, sacrum, everything is there. Slowly and slowly it is coming out. There is flexion. Then there is internal rotation of the head gets rotated. Then there is extension, the doctor releases the head of the child. There is external rotation of the head so that the entire body can come out. And as soon as the baby comes out, this is the placenta, this also comes out. And the uterus gets relaxed. This is the umbilical cord. Now, after parturition, there is another process which is known as lactation. Lactation means the production of milk by the mammary glands. As soon as the baby is born, the mammary glands of the females will start producing milk so that can be fed to the newborn child. The milk that is produced during the initial few days of lactation is known as colostrum. Colostrum is very important for the child. It is the yellowish thick milk which is produced during the initial days of lactation. Why colostrum is important? Because it is rich in antibodies, and these antibodies provide resistance and immunity to the newborn. Now the third homework today is which antibodies are present in the colostrum. Find the answers of all these three questions. So here we complete the entire chapter, human reproduction, a very important chapter because there are many important topics in it. And there are 100% chances that at least one of the topics will come in questions in the board exams. So now your homework I have already given you. 3 questions you have to solve. Along with that, you also have to do all the diagrams of the chapter. You have to write all the processes. You have to make flow charts. And you have to study each and everything of this chapter. If you have any doubts, you can contact me. Until that, keep studying and stay safe, stay at homes. Thank you.